Okay, good afternoon. Welcome back after lunch. So we'll have Yasmine Meroz presenting her first talk. Yasmine is at the Tel Aviv University, and she's going to tell us about the dynamics of rod-like sensory growth systems.
Thank you. Okay, first of all, thank you very much for inviting me here. I'm very excited about being specifically in this winter school, which looks amazing. I envy you from the student side. And also, I love India, so I'm just happy to visit here again. So I guess that today I'm going to do an experiment. Okay, I'm going to mix both the presentation and the board for some of the calculations, and I try to keep it pedagogical. So I'm trying to show. Basically, I want to show here a model for. In a second, I'll define what is this rod-like growth-driven system. But the idea is that I want to go with you right from the beginning of what are the observations, what are the requirements of a model in this case, and how we build the model, and then the investigation of the model, and so on and so forth. And maybe in the end, depending on how much time we have left tomorrow, I'll also discuss what we do in the lab within this context, so it might give you a better feeling. So before I really start, I just want to give, again, for context, just mention in a word what we do in the lab. We look at plant behavior. So I'm a physicist by training, and in the lab we're quite multidisciplinary. We have people with physics from physics and biology, and the idea is that we try to we investigate basic behavioral processes which are ubiquitous in biological systems, such as decision-making, memory phenomena, collective behavior, and so on, but in plants. Now, most of you, when you hear this sentence, plant behavior, might be raising eyebrows because we're used to thinking of plants as this static fellow here, which doesn't look like it's making any important decisions. But my point here is that I think most of this is also due to a problem of perception. Because if we look at time lapses of growing plants, and there are a ton of these on YouTube, you'll see that plants are actually extremely dynamic. They move a lot. They respond to many many different stimuli. And in a second here, you'll see these. This is from a wonderful episode from BBC Planet Earth on jungles. You see these climbing plants, which are moving like crazy. And you can see that there's, I don't know, this plant here is trying to wrap around a leaf, and he sees that it's not a good place to attach. So it continues its search, looking for a better attachment until it attaches there. I mean, something interesting is going on here, right? So if we were living in the same timescale of plants, because I should say that the timescale that you see here is all, the movement is all due to growth. Okay, it's not bending, it's bending due to growth, and we'll go into this in a second. But growth takes time, and that's why it's so slow and we can't perceive the movement. But again, if we were living in the same time scale as plants, we probably wouldn't be looking at them as these static objects rather than living, behaving creatures, let's say. Having said that, now I want to go back to the title of the talk. And the first thing I should answer, I guess, is what are rod-like sensory growth systems, and why do we care at all? So, sensory growth system is basically any system such as the plant that I showed here, which is has a sensory system. Okay, all biological systems have a sensory system, and there is so that they can get information about the environment, and then they respond. Usually through a motor response. I mean, there are many responses, but one of them are motor responses, right? In plants or growth-driven systems, this movement comes out of growth, right? So instead of a sensory motor system, I can call it a sensory growth system. And there are many these systems, but I'm interested in rod-like systems. So these are one-dimensional systems, and I'll give a few examples. One example is neurons. So the small filaments you see there growing, these are single neurons. At the edge of the neuron, you have the growth cone, and basically it is able to sense various chemicals, various gradients of chemicals, and it can grow in a given direction, towards or away of some chemical concentration. And this is a movie of the development of the zebrafish nervous system, which I find very striking. Another example is fungi. In this specific movie, you can see fungi such as these which respond to light, the direction of light, so they have sensors that tell them which way the light is coming from, and they grow towards it. And here you have fungal hypha, and they are able to detect, again, chemical gradients. Okay, but in both of these, you see these one-dimensional filaments or rods, okay, with sensors that can detect some sort of stimulus, and which allows them to grow in the given direction. Another two examples are, of course, plants, because that's what I do in the lab. And here you see two different examples. On the left, it's gravi-tropism. So these growth-driven movements are called tropisms in plants. So what you see here is that they took this pot of, I think it's peas, if I remember correctly. They put it on the side, and now you'll see that the plant is equipped with sensors for the direction of gravity throughout its body, and the shoots grow up, and the roots, which you cannot see here, grow down. You can imagine why this is important, right, to know which way is up and which way is down. And what's nice to note is, at this point, they rotate the pot, and now, so it detected the change in the direction of gravity and grows up. But what you see here is you have this U-shaped organ. So what this tells us is an interesting fact, is that in the body or in the structure of the plant, you basically have encoded information of what its history, what it has sensed throughout its lifetime. Here you have another example of phototropism. So you have the light coming from the left, the crest is going towards the light, and again, they switch the direction of light, and they do this U-turn, okay? So in this talk, as I say here, we're going to focus on plants, and I'm going to show, yeah?
So what prevents these systems from going
sideways?
Yeah.
Gravi-tropism.
No, like, one, it grows in singular direction, right?
Yeah.
But it doesn't grow in width.
Oh, so it's the way that the cells are made. The cells, plant cells are rigid, they have a cell wall, and basically you can imagine that it's more of a square-like cell, and the growth is through, you have cellulose wrapping around, but within a given direction, so it's always this direction. And so basically when it grows, you have the inner pressure that pushes outside, but it can only push in this direction because the fibers can move one apart from each other, and there's a new fiber being attached. So, and since it's made out of many of these cells and they're all oriented in the same direction, it can only grow, well, mostly grow in this direction. There's of course also growth, radial growth, but we'll not touch it here. So what I want to say here is that basically what we're going to do today is tackle models that describe these dynamics, and I'll also try to convince you why it's interesting and important. And although it's the motivation comes from plants, these models are actually applicable to any other system. You can use it also with neurons. You just have to modify the parameters to fit what we see there. And this is, now I come to the question, as I said at the beginning, why do we care? Okay, so there are two answers to this. One, and this is related to maybe what Bill started with his talk yesterday, where he said that in biology, when we want to build something, there is a plan and there is the building, the process of building, and in biology we can't necessarily separate the two. So with plants it goes another step forward. I'm interested in behavior in the lab, right? And in one way of studying behavior is through its manifestation in the movement, because the movement tells me something about the response, and therefore it's a window on the internal information processing process. So in animals, you grow, you develop, and then once you're done, okay, you have your movements, and that's what you study. But with plants, you can't make this separation because movement is due to growth. Okay, so you cannot separate development from behavior. And so if I want to study in the lab behavior, I have to have a very solid model for growth and how it's controlled and how it's regulated. A second reason why it's important or why it's interesting, why I should care, okay, is that actually I think that there's more to this than meets the eye. On the one hand, I will say here you have a really old time lapse. This is from 1898. And you see this gravitropic response of impatience. And you see that there's rich dynamics going on here. It's not just straightening up, it's straightening and oscillating, overshooting, and then finally stabilizing, okay? So it already seems like there's something interesting in terms of the regulation that's going on here. But when you think about it, the fact that you have, I don't know, straight trees is practically a miracle. Why? Because what is a growing system? A growing system means that you're continuously adding more and more, especially in this kind of one-dimensional picture. You have your shoot, and at the top, you're adding more material, more cells, and more cells, and more cells, and more cells. And these cells are also elongating, right? And this is pretty much similar to having a lot of pancakes and adding more and more and more and more. But as we all know, at some point, this deck of pancakes will topple, right? You have to have people holding it. Or you can imagine when we build large buildings, skyscrapers usually have bigger and bigger radii, let's say, of the building. And of course, you have a ton of issues of construction you have to think about in advance. But this is all happening as it goes, right? It grows and it responds to, like you can shift the plant on a side and it will be able to detect the direction and still grow up. So this is absolutely not trivial. And actually, within the lab, we're part of this, almost a year ago, we got funded within this large collaboration of 9 labs across Europe, called, the project is called Growbot. And there the idea is that basically this project proposes a new paradigm in robotics. So most robots, if you know all the movies from Boston Dynamics, the company, right? There are these really cool robots that do whatever you like, but they're all inspired from the animal world. And this means that the movement comes from self-propulsion. Right? They have legs or wheels, basically. But then it means that they get stuck the moment that you put them in, let's say, ruins or some unstructured area. So what Growbot says, okay, maybe we should look for inspiration in a different area. Let's look at plants instead. And growing plants have no problem with obstacles or crossing empty spaces and going through cracks. They're really good at that. And so basically the paradigm that Growbot is proposing is to make a plant-inspired or climbing plant-inspired robot, which moves due to the addition of material. How? It's a different question. This is complicated. But my point is that actually when you go, kind of, Feynman said that what I cannot build or create, I do not understand. I have to say that when we go and I talk to these roboticists and they ask me, okay, we need to write this control model for the robot. And then we see that, wow, even just standing up, and that's it, is difficult. It's not trivial at all. Okay, so this is a bit of a parenthesis to the whole issue here. I'm just trying to say why, to point out why this problem is involved. Okay, and now the question is, given everything that I've mentioned here, I want to understand the dynamics and the regulation. What's going on here? Okay, is it something is the regulation centralized? Is it distributed? There's no brain here, right? So what's going on? And I'm gonna break down the talk into these different parts, and then we'll see how much time we have left. I'm assuming that this will also go to the next talk tomorrow. So I'll start with understanding, first of all, maybe some biological perspective of perception. We're going to talk only about gravitropism here, just to keep our talk focused. So I'll explain a bit about the biological aspect of the sensing and the signal transduction on a very basic level. Then I'll describe a bit the response kinematics, so you can get a feeling of what we're dealing with. Then we're going to discuss mathematical modeling. So we'll construct a model with basic requirements and assumptions. And we'll see how it works, if it works well or not. And then we'll add an extra complication, which is the influence of growth on regulation. So let's start with perception and the response kinematics. very briefly, I'm going to do a lot of hand waving right now in this section. What you see here is that, yes. Okay, so this is a cross-section of a shoot, and please stop me with any question. Anything is not clear, please ask me. This is a cross-section of a plant shoot in the area which grows here, the elongation zone. And here you have a cross-section of this external part. So this is the endodermis, and this is the epidermis. And these box type of cells, which I mentioned before, right? They're kind of quite squarish, rectangular. So the one that's marked in red is shown here, represented graphically here. So, and this shows us the sensor, basically, the gravity sensor. Basically, it's a quite elegant idea because it's based on mechanics. You have these amyloplasts, these circles here, and these are just sacks of starch, basically, and they sediment. That's all they do. So now if your plant was straight and you put it on its side, so now what happens? All of these amyloplasts will sediment to the new bottom of the cell, right? And when they touch the cell, basically they tell the cell, okay, this way is down. And then there's some sort of signal transduction which is not well understood yet. And here you have, there's a difference between the shoot, the aerial part, and the root. In shoots, these cells, the sensing cells, statocytes, appear, they're basically along the whole shoot, whereas in roots, they're only, they're focused only at the edge, at the tip, as you see here. Okay, so this is the tip of the root, and they're basically the ones that are colored in this black color. Those are basically the amyloplasts that are colored. You see that they're quite here, they're all only at the bottom. Okay, so their sensing is only at the tip, whilst here sensing is local, it's everywhere. Now, I'll take this one step further. So what occurs when these sensors sense something, that something has changed? So you have here your plant, and there's a growth hormone called Auxin. And Auxin is like this magic hormone. It does everything in plants, but specifically in this case, in the regular setting, it will be homogeneous, okay? So if it's homogeneous, it means that the plant grows straight, okay, homogeneously, yeah.
So what is actually detected? Is it the number of amyloplasts which are in contact with the membrane, or the total number of...
It's not known, actually. There is a lot of speculation about this. What has been, one of the recent findings is that apparently it doesn't sense the magnitude of the force, the acceleration, but only the inclination. So that's one of the last findings in the area. As for the actual what is being sensed, I'm not sure. There's also, of course, there's reason to believe that it's not just the amyloplasts. I mean, I'm really oversimplifying here what's happening, but basically it's not only the amyloplasts that hit the bottom of the cell, but there's also the cytoskeleton here. And they fall along, they pass through the cytoskeleton, so that seems to be also involved. And it seems there are mutants without amyloplasts, and they still sense gravity. Not as well, but they still sense. So there's obviously another missing ingredient here, other organelles that are aid this sensing, but it's not known. You mean in the area which is of the shoot or of the root? I think they're no longer active, because in any case, whatever is outside of the growth zone cannot change its direction. So in that sense, from an engineering point of view, it doesn't help that there is a sensor if you can't do anything about it. Okay, so what happens now? I just want to finish this line of thought. So you have basically Auxin, which is homogeneous, and you grow straight up. And here are the sensors, of course. Also, photoreceptors are on this side if we're talking about phototropism. Now, if I put some signal from the side, whether it's putting it, tilting it, or whether it's light or whatever, what happens is that basically Auxin is redistributed to the back, to one side. Okay, so now what happens? You have a lot of Auxin, of growth hormone on one side, and this will grow at a high rate, and very little auxin on the other side. So this will grow at a slower rate. So if one side grows more than the other, you'll get this bending, basically. Okay? So all the movements that you see, that you saw before in the movie, they're all, if they're growth-driven, they're all through this differential growth. Okay, that's the main story behind everything that we're seeing there. Now, another thing to note is, and this has to do more with shoots, of course, is the locality, okay? What I mean is that sensing is local. So we saw that there you have sensors throughout, so every different point in space can sense gravity, but also the response is local. And this experiment, whoops, it's not what I meant to do. This experiment, which is from '97, did an interesting thing. You have here a shoot, and this thing here from the side is basically a magnet which manages, the magnetic field is very centralized or focused around the tip of this object. And what they show is that, and this magnetic field is able to basically move the amyloplasts to the side, okay? So it's basically messing with the plant and telling him only in this area where it's touching, hey, here, down is this side. And you see that it's responding, okay? So this shows us that you don't need the whole plant to sense gravity. It's enough that it's a local process, okay? And this will be important for when we go towards modeling.
So how is it with the magnetic field? What are the particles responding?
It's moving the amyloplasts. I don't know how exactly. I'm not sure what they did so that the amyloplasts will respond to it. I will have to check and tell you. But the take-home message here is just the fact that the response and the sensing is local here. Say that again, sorry? I don't know. I honestly use this just as a reference for the fact that it's local, but I can check and I'll tell you. Okay, and now I just want to give maybe some intuition as to what it means to work with gravity when looking at plants, and how experiments that we do look like. And also to give you, this is a short movie from collaborators, Bruno Moulia in France. Actually, what I'm showing today is all references from a few of their papers. So it's Bruno Moulia, Yoel Forter, Stéphane Douady, Renaud Bastien, who's the first author in all of them, and Thomas Bohr. So in this movie, what you see is the whole experimental setup that we're also using in the lab, but also it shows you the statoliths moving live. So that's why I think it's nice to see this. Okay, so there's a microscope. This is Bruno, who's saying a lot of smart things. And now you see that this microscope, okay, here are the cells, and these dots are the amyloplasts. This is Ugo, the postdoc who's working on this project at the time. And what you'll see is that what's nice about their setup is that it rotates. So now there's a live tissue in there of plant. They rotate it on the side, and now you'll see in a second the sedimentation. Just a second. There you go. And there's a lot of nice work about looking at this as a granular medium and looking at it, yeah, this is the physical representation of this. And now the point here is that basically, since these amyloplasts are sedimenting within the cytoplasm, which is a viscoelastic medium, it means that you can do a trick. So what you see, this rotating machine here is a clinostat. It's a fancy name for a very low-tech piece of engineering, but it's very useful. These are wheat coloptiles, the tiny green things that you saw in there. So the idea is that you rotate all of the plants in a low RPM, okay, rounds per minute, so it's slowly. But if there's this sweet spot of RPM where basically these amyloplasts never sediment, they're just stuck in the middle, right? If you do it very quickly, like in a washing machine, they'll hit all of the walls. If you do it too slowly, then they'll just tumble always on one side. But this is basically somewhere in between, where they're always in the middle. And so this messes up with our sensors, because basically it tells them it's canceling out gravity, okay? So it's a cheap way instead of sending them to space. But it means that we can do pulses of gravity, right? Because we can have, which we can't do if we send to space, right? We can have signal, 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 okay? Then we start rotating, no signal, no signals, no signal, and then we stop it, signal, signal, signal. So we can do nice dynamical patterns, which is nice. And then we have these time lapses. We take pictures of the plant shoot, and we do image analysis from the form. And as I said, we need to look at since we know that the movement or the growth dynamics tells us something about behavior, right? So what we do in the lab generally is all of our experiments around the idea that I give a stimulus that I know, that I control, I watch, I observe the dynamical response, and from this I can infer, kind of reverse engineering what's going on in the black box that kind of processes information from the environment, okay? So I hope this gives a bit of a better idea of how we go about this in the lab. Okay, so now I want to move to the response kinematics. Up until now, I basically talked about the perception or sensitivity, sensing. Yeah?
How does that go? Is it parallel to the gravity? In case of the gravity was cancelled out.
Yeah.
How did the plants?
They continue to go sideways. Yeah. They grow in because Auxin isn't redistributed, so they continue to grow in that direction. And yeah, actually, I didn't say, but the dynamics that you saw, that it was going up and down twice, the wheat, that's exactly because we were giving pulses of gravity. So it was rotating, there was no gravity, so it was going straight, then we stopped it, so it goes up, and then we started rotating again, so there is no more gravity, so it goes back to going straight, and so on. So dynamics. I want to show here two of these experiments. On the left, it's a wheat coloptile. On the right is Arabidopsis thaliana inflorescens. Arabidopsis is kind of the E. coli of the plant world, okay? Because we know all of the genome, and there are a ton of mutants. Although it's a very annoying plant actually to run experiments on, but that's a different story. Now I want you to look at the dynamics. This is one. Okay, so you see that it curved and straightened up, and it reaches some sort of steady state in terms of the geometry. Now let's look at the other one. It goes up, it overshoots, goes back, slightly overshoots the other side, but now it's reached its steady state. Okay, so we see that there is a variety. First of all, the dimensions there's a big difference here. And also the types of dynamics. Does it overshoot? Does it not overshoot? Okay, there's a variety here. So now we want to parameterize. We want to say something a bit more quantitative about these movements. So how do we go about it? In order to parameterize this problem, I will use one variable, which is theta, the angle, the local angle. Okay, so first of all, if this is my organ, okay, S is a variable that goes from 0 at the basis to L. L is the length of the organ. Okay, this just goes along the median line here. Okay, it tells me the position along the organ. And now at a given point S, I can look at the angle from the vertical, at a given snapshot, of course, okay? And this defines now this local angle. So theta at point S at time t. So you can notice, for example, that here theta is almost 0, right? Because you're practically vertical. Whilst here, it's clamped, okay? The boundary condition is that it's 90 degrees at S equals 0. And another thing that we can look at is the curvature. So the curvature is just the derivative of the angle over S. So it just tells me if I move from this S to another S, which is close by, how much does theta change, right? If the curvature is big, then theta changes a lot. So it means that you're really curved. But if you're completely straight, then theta doesn't change at all, and kappa, or the curvature, is 0, right? And although it seems from the mathematical point of view, this is trivial to say that theta is the integral of kappa, but the point here to notice is that the angle of the tip, for example, okay, is basically a sum of all, or an integral of all the curvatures going up the stem until this point, okay? So a small change in curvature down here can have a very big effect on the angle on some other place. Okay, so that's just something to keep in mind. Right, so now what we're going to see, because now we want to go, okay, we said that we want to describe I showed you before the dynamics of two different plants and said, okay, it looks like one oscillates, one doesn't. What's going on here? So now that we have these parameters, yeah?
Is there some sort of overdamped, underdamped mechanics?
You'll have to wait and see. Yes, okay, but we'll talk about it in a second. Yeah?
You're assuming planar shapes?
Yes.
Things that don't go out of the
Yes, yes. So, okay, that's a good question. In general, we see that all these tropisms are always in the plane, in the plane, because the stimulus in this case also goes from infinity, right? It's gravity, you have a parallel field, and you're always just moving inside the plane. There's no reason to move outside. Although I should say that plants do have inherent internal motion, which is called circumnutations. This is kind of a rotational movement. So if you look at a plant with no stimulus, you'll see it rotating a bit. But we disregard this here. We kind of look at it as some sort of noise, let's say. Okay, so now what we have here, this is taken from an experiment. Okay, we did image analysis, and then you have the form of the shoot. This is the wheat coloptile that you see here. And now we color-coded the curvature. I didn't write it here. No, so this is the curvature. Blue is straight, as you can see here, and red is very curved, okay? And you can see it here along, so this is a local curvature. It's color-coded here. And you'll see here the movie of the wheat, and here you'll see a spectrogram, if you will. Basically, you'll see the x-axis is S, so this is the basis. This is the flip picture, basically, of the movie. Okay, this is the basis, this, and this side will be the apex, which is this. And this is time, going from top to bottom, and you can see the evolution. So let's see how it looks like. So is there anything special or interesting, or is there anything that you notice here about the curvature with time? Very well done. Yes. Okay, so it seems, maybe I'll show this again, okay? If you look at the curvature, you'll notice that there is initially, the curvature is well distributed, so everything curves, but very quickly the tip starts to straighten and the curvature is moved down, okay? And it's focused at the bottom. So most of the plant is actually straight at the end. So you can look at it again. See how it's focused at the end. And here you can see it, of course, in the spectrogram very clearly. At the beginning, the red spot, the red blob of curvature is almost along the whole shoot, and then it's only on the bottom part. Okay? Yes, so the x-axis is the mirror picture. Actually, no, it's not. No, it's not. Right, this is the bottom. Yes, sorry about that. With the different figures are always with different axes. Sorry about that. And you see here that the length, of course, is growing. Okay, that's something that we'll have to discuss also. Okay. Yes, there are some oscillations, but right now we're just considering the main features here. And we'll see in a while why this is important. Why the point that you have the curvature which focuses to the bottom? Yes. So of course it's a noisy system. So you'll have in any case some small oscillations. Some of them will probably be due also to the circumnutations that I told you, though here they're not pronounced at all, but they're probably due to that. Yes, you see them mostly at the beginning. Okay, so now I just want to show you some other figures from completely different plants. Okay, so this is what we looked at a second ago, the wheat coloptile, right? This is what I showed you here. This is for the Arabidopsis inflorescens. Inflorescence just means what developmental stage it is, okay? And you can see the scale bar, right? This is significantly bigger than this. And here you see a whole tree, okay? This is a couple of meters, I think, a poplar tree. This is data that was used, older data that was used just for the analysis. That's why it's quite noisy here. But what you can appreciate here is that though you have a very large variety of scales and it's completely different plants, right? Also in different developmental stages, right? This is the tree, the growth is, there's tension. That's what the tension in the wood is what changes the direction, okay? It's not exactly the same mechanism as here, which is differential growth. And yet, you still have the same shape, right? You have this general curving at the beginning, and then this the curvature moves is focused to the basipetal straightening, right? So the apex is starts to straighten out, and the curvature is focused at the bottom, okay? So this is a very striking universal feature, if you want, of these dynamics. So let's start to model this thing, okay? Yeah.
How did the overshooting displayed in the graph here? The second case, you said there's an overshooting and overshoot back.
Well, here we see only curvature, not the angle. So it's hard to see here, because the curvature is already the derivative of the angle, so it's a small fluctuation. You would see it better. I mean, it's something that you should probably see in the details here, but it's hard to see. But if you were to look at the angle, you would see it better. Yes?
Is it surprising that the overall length change over time is also very linear?
No, but I don't want to go into this now because there's a whole section of growth later, so maybe it would be better to disconnect the two for a second, okay? Okay, so I want to go with mathematical modeling. So what are the requirements of our model? We want it to be simple and parsimonious, so a minimal model, so that it will be intelligible. What do I mean by that? I don't want something with 1 million variables and 1 million parameters that I have to fit, because then, as you know, you can fit an elephant into 100 parameters, right? So I want something that's very minimal. 5 parameters is enough for an elephant, there you go. So I want something which is minimal. And more than anything, one of the reasons is that this allows us to identify, okay, this term is associated with this, which I can measure experimentally. This term is associated with this, and this allows me to get a better understanding of what's going on. Okay, and you'll see that when we build the model this way, we can build a model, look at the dynamics that we predict, and then go to the observations and see whether it fits. And if it doesn't fit, we say, okay, something is missing. What's missing? And we go back to the writing board and develop the model again. So I want to go through this process with you. Okay? Okay, the second requirement is that we want to reproduce, we're going for a qualitative model, okay? So we just want to reproduce the striking phenomena that we see in the dynamics, okay? This initial curving and then the straightening out. And we want it to be robust and universal. What do I mean by that? Just as we saw that with plants, you had this large variety of scales and of completely different genotypes and different mechanisms of differential growth, and yet we see the same dynamics. So this model also has to be able to capture this large variety. So this is what we require from the model. Now let's go with the assumptions. What do we assume before we start writing anything? We said local sensing and local response, right? So this is one thing. Another thing is, okay, so now let's look at the two different ingredients. We have the sensing is one part and the response is one part. So let's say that the response, the dynamic change of the form, can be basically, it's encapsulated in just looking at the change of the curvature in time, right? Which is this derivative. Kappa is the curvature, and I look at how it changes in time. This will tell me something about the dynamics. Okay, this expresses the movement. And the stimulus is gravity, right? Right now, we say that gravity is the only stimulus. And how do we sense gravity? Okay, when I'm like this, or when the plant is straight, it doesn't sense, like, it's straight. Everything's fine, right? So, but if you put the plant now at 90 degrees, then you'll see the change. So what I'm saying here, basically, is that gravity is sensed via inclination, okay? So this is our theta. So now let's construct this model. So we start with the response. Response or movement. Can you see this? Okay. And we're saying that this is a function of the stimulus, gravity, right? So we said that this will be represented by theta. So it'll be some function of theta. And where do we see, where does the fact that we said that everything is local, where does this come into this picture? The fact that we have S here, okay? So for every point in space, S, along the organ, this thing occurs. And the angle will be different at different points, right? So this is where the local part comes to place. Now we say, okay, we don't know what this function is. Let's expand this into a Taylor series. Yeah. How do I what? Sorry?
Inculcate the growth of the.
Okay. At this point, we're going to assume that the length is fixed, and we don't look at growth yet. We'll just look at the dynamics. We'll discuss why this is right, why we can do this in the end, and we'll also add actually growth in the next section. But for now, we're simplifying the question. Okay? Since also, as you can see from the movies, I don't know if you noted, but you have a lot of interesting things occurring at the beginning, and then once you're at the stable state, you just move up, but that's it. So to some extent, you're interested in what happens at the beginning. Okay, but it's not a good answer, but I promise a better answer afterwards. Okay, so we can express this as a series. So it will be some kind of beta 0 plus beta 1 theta st plus beta 2 theta squared and so on. Great. Now, we know that this problem has symmetry. What do I mean by symmetry? If I look at my plant, this is my plant, and this is theta. If I look at theta this way, or if it were the other way around, like this, right, this is minus theta, but it's the same. I don't care which way it's directed, right? So the same equation should work both for theta and minus theta. And since curvature is the derivative of theta, so the same goes for curvature, right? Okay, so what does this mean? Let's put now, substitute everything with a minus. Okay, I have minus kappa dt, beta 0 minus beta 1 theta plus beta 2, theta squared minus beta 3 theta to the third, and so on. And now if we compare the two, we see that all the even powers will have to go to 0, right? So because, whoops, yes. If I multiply all of this by minus, right, so it will be minus this, minus, 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 right? So now I can compare because this is equal to this. So now this is okay. Minus. So this can only be 0, right? Minus beta, if minus beta equals beta 0, it has to be 0, so this drops. And the same goes for this. If minus beta squared equals beta squared, it must be 0. So I'm left only with the odd powers. So even powers drop. So what am I left with now? Theta 1, theta plus, I'm dropping the s and t just for the sake of simplicity. I mean, for my ease. And so on. Okay? And since we like, yeah. Yes. Okay, and now since we want our lives to be easy, we'll just take the linear approximation, okay? Why did I do this? I don't know. I can already tell you that the way that we like to write this is like this. Okay, and here at this point I will already say, when I said here that I don't know what the function is, I was semi-lying, because actually there is a lot of work that shows that, that actually the theta is sensed as a sine law, okay? So actually, this is exactly right. We have all the odd powers, which is a sine, okay? So we could use, instead of this law, but we'll see that the linear approximation is actually really enough for to do what we want to do here. I mean, it shows the same dynamical traits, but it means that we can solve stuff analytically. So keep in your mind that the more full description is a sine law, but we're using the linear approximation here. Okay? Is there anything else I wanted to say? No. Okay. So let's see how this model works. Okay, what it does for us. So on the right we have our solution. Since later the solutions will be really ugly, maybe here I'll show you quickly the main idea of how to solve these equations, but I won't go into technical details. I just want to show this. Okay, so our equation is, yes, T, yeah. I'm showing now here, but with a little bit of hand waving because I don't want to go through all the details. What time is it? Okay. Okay, so I'll really do this quickly because I don't want to waste too much time. Remember that kappa is the spatial derivative of theta. So actually what's written here is d kappa dt ds, d theta, sorry, and theta here. Okay? And now what we want to do is a Laplace transform. So a Laplace transform is like a Fourier transform, right? And basically it will look like this for those of you who are not sure about it. So we're doing the Laplace transform on S, the spatial term. Usually it's done on time, but here we're going to do it on S because S goes from 0 to infinity, okay? So it's done on S, and the variable will become P. It looks like this. Okay. And now what we do, if we do the Laplace transform on both sides, okay, notice that the derivative of time doesn't care about everything else here, right? Because the integral is over S, okay? We'll only care about theta here. So it will look like this. Okay. Actually, you can also assume that beta equals 1 here for simplicity, okay, by renormalizing everything. And now you get, so here I can take the derivative of time outside of the Laplace transform, as I said. So it will be like so. Okay? So it's just the d to dt of the Laplace transform of the derivative in S. And now this will look like this. So with derivatives, you have p and theta. Theta hat is the Laplace transform minus the derivative of theta at S equals 0 at time t. Okay. We know, if you remember, I said that the angle at S equals 0 is constant. It's clamped. So the derivative will be 0. So this drops. So I'm left with just this. Okay? So we have, sorry, without the beta, because I linearized this. I normalized this. Okay? So now I have basically the derivative in time of theta with this parameter p equals theta. So we know how to solve this, right? I'll rewrite this. D to dt. Sorry, I'm jumping between notations here. Of theta over theta equals 1 over p. And this gives me theta okay? So this tells me that the Laplace transform, this I can solve. It gave me that the Laplace transform of theta has this exponential form. And when I do the inverse Laplace, I get this. Okay? This is just a table. Now, what you see here, J is the Bessel function. So if I would take one thing home from this thing, because the actual calculation maybe isn't so important, but it's a nice trick for partial differential equations. The Fourier and Laplace transforms can sometimes change, they change derivatives into just multiplications, so to an algebraic equation. That's very useful. Okay, so let's look at this solution. How does it look like? So on the left, we have this sort of graph like before, the curvature. So this is time, from time 0 to time 5, whatever that is, okay? So you see that at the beginning, it's completely straight, and then you get this curvature at the top, but then you see all these oscillations here, which you can see very well in the video. Okay, maybe I'll try to show the video again. So it overshoots, it doesn't straighten out, like we saw in the plants, right? It continues to oscillate, and actually here at some point the simulation ends, but this will continue to oscillate forever. And we can see it from this Bessel function here, okay? So this is not good, right? Because we're not reaching a steady state, so something is missing here. What's missing? So, yeah.
The knot is the initial angle.
Say it again?
The knot is the initial angle.
Yes. Yes, yes. So let's try to understand this. What you see here, this is a nice figure that was done by someone who discussed the PNAS paper that shows what we're going to show in a second. The point is, okay, you have gravity is sensed everywhere, right? This is the initial state. Gravity is sensed everywhere, exactly the same direction, okay? Now you start straightening out or curving, and now at different points, the orange arrows represent the component of gravity that is being sensed because you're curved. So you see that in the top part, where you're practically vertical, you sense almost nothing, but here you sense a lot at the bottom, okay? So what happens is this. Basically, at the bottom here, even though this has already arrived to the vertical, at the bottom I continue to curve because there's the component is very big, and I continue to curve even more and even more, and I curve so much that the top overshoots. And now with the overshoot, you have the same issue. Okay, you continue to curve. So basically, the fact that you're sensing the inclination everywhere, and that's it, causes this kind of oscillations. Okay, and you have the lever arm effect because every small change of curvature at the bottom moves everything, right? And that's why you get the overshoot and then another overshoot and so on. But what we saw, again, was different, right? This is just the shots of the first black and white movie, the overshooting one. So true, it does oscillate, but after and overshoots. But all the plants that we considered here, they all arrive to a stable state at the end, even if they oscillate. So there is something stabilizing that's missing, okay? And if we go back to our movies here, so on the left is the movie that we saw before, where we saw that there was, and here there's just an older paper which shows the same idea. You have the general curving, and then here comes the interesting part. You have this straightening out issue, right? Here, the arrows show the curvature. Never mind, you see it here as well, right? Yeah?
In the figure on the left, is the shoot growing all along its length? Or only
Yes. No, all along. So basically, if you look at just the initial stage where everything curves up until here, or up until here, if you will, that's similar to just the regular model that we showed where everything is sensing gravity, okay? But we can't explain this with our current model. So the fact that it's straightening out, this has been called autotropism, and we like to call it Bruno Moglia et al., and I follow their work, so I add myself to the we. They call it proprioception. Okay, this proprioception meaning just like we use proprioception in people, you know, where we are able to sense where our body is, if my arm is straight or not. So in the same kind of idea, the plant senses that it's bent and it doesn't like it, so it straightens out. Okay, because for various biophysical reasons, it's not good to be overbent. Okay, it prefers to be straight. So this is something that's missing here, and you can think of this as a restoring force. Yeah.
Straight meaning zero curvature? Or?
Curvature, not angle. Yes. Yes, I forgot what I was about to say. Okay. So yes, restoring force. Because why restoring force? Because gravity calls me basically to bend, right? Or any stimulus, right? There is a stimulus in one direction, I want to bend towards it. But this proprioception thing means that I don't want to overbend. So it's a restoring force in the opposite direction, let's say, okay? These can be two opposing signals. So let's see now that we know that we want to add, okay, I already showed the equation. Let's go back and let's say that we didn't see the slide. And let's go back to the construction of the model. Maybe, yeah.
We analyzed the situation where we had essentially kept the length of it.
Yeah.
We have done this regime analysis of what will happen. But we see the stabilization, here we predict output. We see the stabilization really in the growth part. So,
Well, all the dynamics are in the growth part, because once you're not in the growth zone, we'll discuss this later, but there's an area called the growth zone, and whatever is below that at some point, it doesn't grow anymore, the cells do not elongate anymore, and there's no addition of material, and that's fixed. It can never move, it can never respond. So if we're anything that, in any place where we consider movement, it's growing. So right now, in this case, in these models, we're simplifying, we're saying that everything is growing. Like the growth zone is the whole organ, but we're also disregarding the elongation effect. I mean, we will add that in a second, but for now we're disregarding it. We're assuming that L stays the same. I promise that I will answer your question in a bit, okay? So maybe I'll go to this side of the board so we can keep the other side. So now what are our assumptions? We still have the gravity signal. So, okay, everything is still local, fine. We still have the gravity signal, okay? Gravity, which is sensed through inclination. But now we're saying that we also have proprioception, this restoring force. proprioception. And as you're asking, what is it sensing? It's sensing its curvature, right? So basically, it will be something, a function of kappa, not of theta, okay? So now if we want to go back, as we did before, we have d kappa dt, the movement, will be some function of both theta, which is gravity, and kappa, okay? Now again, I can go and expand this, okay? So I have, like before, beta 0 plus beta 1 theta plus blah, 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 all the powers, plus gamma kappa, gamma 1 kappa plus gamma 2 kappa squared plus blah, 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 and all the combinations in between. So phi 1 theta kappa, so this is just a multiplication of the two, and now third order. So phi 2 theta squared kappa plus phi 3 theta kappa squared, and so on. Yeah?
Sorry, so the model of the perception is a function, but the reality is that action or the absence of action. Is it like when you don't respond to gravity, like the growth doesn't respond, then you have a perception, or is it an actual phenomenon? Something happens as a result of the perception?
Okay, you're saying that in gravity we said that there is a whole process of signal transduction and so on, whether proprioception is a mathematical creature or a biological creature. So it's a biological creature. It's not well understood. Recently, there's some evidence that shows it's probably connected, that when you look at the dynamics of Auxin, so you look at these sort of straightening experiments and you follow the distribution of Auxin. And you see that initially Auxin is redistributed, as we say here, but actually after a while, it seems to change direction. And this might be associated with proprioception. I should also say that in general, plants sense mechanical stimuli. Okay, so they're able to sense changes in their structure. So you might imagine that when you're bent, there is some internal forces or something that activate a lot of hand waving, yeah. But it's not well understood, but it is a biological, it's not just a mathematical creature. Okay, so we have this equation now, and like before, okay, we have symmetry, so all the odd powers drop. Yes, and we're left so we're left only with the even powers, and like before, we want to use the simplest linear approximation. So we're left with what was on the board, the kappa. Sorry?
I thought you want to retain the limit. You want the theta minus and theta to minus.
The odd, yes, I of course mix this up. I am very sorry. Sorry about that. Yes, the other way around. Okay, yes. So I will now only take the first orders. So these fall because there are higher orders, and I'll only take the linear terms. So these two. So it's minus beta theta minus gamma kappa. Okay. So now this is my new equation. So let's see how well it does. Okay, this is how the analytical solution looks like. If you're interested, you can read it. It's a bit longer and more tedious. But what's the interesting part? So, oh, yes, here. So basically you have here, again, these Bessel functions, which are, again, oscillatory. But what's interesting is that you have here this exponent that decays with time. Okay, so this already tells us that decaying is nice, okay, if we want something to stabilize. So let's look how the dynamics look. And I'd say this looks much better, right? you have this initially, everything is curved, right? There's this general curving, but at the end, you see that or very quickly, the curvature is concentrated at the bottom, as you see here. And indeed, as you can see in the simulation here, it stabilizes. So you do have oscillations. Actually, in this movie, I didn't notice if it's with oscillations or without. Yes, it overshoots once, maybe twice, but it stabilizes. Okay, so that's good news. And in fact, if we now compare this, okay, this is experimental data. This is the current model that we're discussing. This is a previous model. So this is rubbish, obviously, but this looks not bad, okay? So let's continue with this model and see where it goes. First thing that we can do is to try and, okay, when we have a model, something useful is always to look at the steady state, okay, if we have a dynamical system. So if this is our equation, we want the steady state, so it means that the derivative, the movement goes to 0, okay? We just want to look at the final form, okay? So then we have basically just this side of the equation that goes to 0, right? And here I already changed, instead of kappa, I put the derivative in space, just because then I can immediately see that this is a differential equation which I know how to solve. And again, I get this exponent. So theta, the form, the inclination as a function of s, right, along my organ, is this exponent. And here I have both beta and gamma, okay? Remember, beta is basically the sensitivity, if you will, okay, of the gravity, the gravitational stimulus, how strongly I respond to it. And gamma, over here, is how strong proprioception is. Okay, these are two different sensitivities of two opposing signals. And I can define LC, which is a convergence length, so that it will be encapsulate beta and gamma, and then I can, because then I can, you see that if S is a spatial parameter, then LC also has to give me distance, right? So this also tells me the dimensions. I mean, it works with the dimensions anyway, of course. Okay, so what is this convergence length? This now means that I can go take my steady state. So this is experimental work from before. This is a steady state. It doesn't move much after this. It only grows, of course, but it doesn't, there are no more, kappa doesn't change anymore. And I can, okay, I have to identify where the growth zone begins. So this is a dotted line. Here it's fixed. You see that it doesn't change, it doesn't curve. But from here onwards, you see that there is dynamics, and here's where stuff is growing. So the growth zone is 6 centimeters. It's the yellow line. But more importantly, now I can go and fit at every given point S, I measure the angle, and I plot it here. So now I have for different S's, so I don't know, S equals 10, okay? If there's a red dot here, this is over, they're like, I think they did 60 plants or something, and measured the angle at that specific point. And what you get is this nice exponent. They fit it to E minus S over LC, and they can extract LC. So the length of, the convergence length is 1.3 centimeters. Okay, so this tells me how curved, over what length do I see the curvature. And here it's pretty small. You see it's a small area here. Okay, I can already tell you that for the wheat coloptile, which was much more curved if you remember, this will be much larger. Okay? And this tells me that from the steady state form, I can say something about the ratio between beta and gamma, right? Or between the sensitivity to the stimulus versus how strongly I perceive myself, my own form. Okay, so it tells me something about this balance. Yeah. Yellow, yes.
Is that something which you know biologically, or can't it be that you have this initial region which doesn't bend? As actually a mechanical property?
There's also, of course, changes in mechanical properties, like in systems that become woody systems, so you have a bigger bark, but irrelevant of that, still, irrespective of that, you still see that there is, in these cases, what we're considering, these are systems where it's safe to say that there is no growth. You can see just that cells don't elongate anymore.
Because even the plots in the beginning, when you showed all the related stresses, like you could see that the region where you are in manual computation of this was not at the absolute bottom, but it was always slightly above that.
So growth isn't homogeneous. The growth rate isn't homogeneous. You'll see that, I will show that later when we talk about growth. It's not a step function. It's a kind of, the growth rate is a Gaussian, if you will. Okay, so at the tip it grows very little, like it elongates very little. And outside of the growth zone, it doesn't elongate at all. In the middle, there's some kind of maxima. So it's of course not homogeneous. But for the sake of this model, again, we're looking at the simplest model. We can add everything afterwards. We can add 3D, we can add elongation, we can add elasticity, we can add whatever you like. But here we're just trying to really take the minimal ingredients. Yeah.
So continuing on this point, does that impact the model on the boundary condition?
Oh yes, the initial, the angle to which you're clamped. Yes, so you can generalize this. Here we're discussing also that apart from the clamping, that's one thing, but also the direction of the stimulus. I'm here always assuming that gravity is from this side and it's with the vertical. So all the angles are compared to that angle. But you can imagine with light or, I don't know, another setup where the stimulus will come from a different angle compared to your reference frame. And you can just add, instead of theta, it's theta minus theta p. That's a constant angle of the stimulus, as well as the boundary conditions, which also come into play. But they don't change the type of dynamics that you see, in any case.
My guess was that instead of fixing the theta over there, why don't you fix the derivative of the theta? So, I mean, it always starts.
Also, so the bound, actually, yes, I didn't mention anywhere the boundary conditions and initial conditions. In the gravi-tropic experiment, the boundary condition is both, since you have theta and the derivative of theta, you need two boundary conditions. So you have theta is some theta 0, here it's 90 degrees, and also kappa is 0 at s equals 0. You're completely right. Okay, now let's discuss characteristic scales. We already discussed two length scales, which have to do with the shape of in steady state, right? So we have the convergence length and we have the growth zone, okay? Now, let's talk about time scales. There are two time scales that have to do with the dynamics. One is the arrival time to the vertical. So if I run one of these experiments, and let's say the Arabidopsis, you saw it overshoots, right? So I can measure what is the time that it takes until it reaches the vertical for the first time, before it overshoots and so on. That's the arrival time. And the convergence time is what is the time that it takes for me to reach steady state, okay? And I find here, and this is also extracted from the equations, but I won't go into this right now, but they're related in this form to so the convergence time has to do with proprioception, because that's a restoring force. So you can assume that even in the absence of gravity, you still have this restoring force, and that's why that sets the convergence time. And arrival time to the vertical, this is more associated with the stimulus, right? The gravitational stimulus. Okay, so let's say that we have these scales. I can now look, I want to look at dimensionless values, parameters, right? So I can look at, divide these two and get one parameter, and divide these two and get a parameter that will hopefully tell me something about the system. So when we do this, so this tells me something about the kinematics, right, to the movement, and this tells me something about the shape, right, because this is time and this is space. And when I substitute all of these values, what I find is that it's exactly the same, which is, I find, super surprising. This tells me that since these two numbers have to do this BL, right, the dimensionless, the ratio between these two lengths, which has to do with the steady state picture. So I know nothing about the dynamics, but I see just the final form. This gives me information about how the dynamics should look. right? Because how the dynamics should look is related to these timescales, okay? And they're both encapsulated in this dimensionless parameter B. And here I give some examples. These are the steady states of simulations with different B values. So you see that when B is low, you get something closer to the wheat collop type, right? Where it doesn't reach the vertical. And with a high B, you do reach the vertical, and most of your organ is actually vertical. And for example, what we looked at before, the Arabidopsis had a B of 4.6, so somewhere over here, okay? And here I have a nice video that shows the dynamics here, and we can try to understand the meaning. So this is the low B, medium B, and high B. Well, you also have a bit of an overshoot and small oscillations that go away. So how can we understand this in terms of the different scales? So let's say with a low B, okay? In this case, so here I remind you the ratios. With a low B, it means that the time for convergence, the convergence time, is smaller than the arrival time. So if this means that I converge before I'm ever able to arrive to the vertical, right? As opposed to here, this means that the convergent time is much longer than the arrival time. So I arrive, and then there's all the dynamics at the end, but I already arrived to the vertical, and only after that I converge. Or I can look at the length scales, right? This tells me that in this case, the growth zone is smaller than the convergence length, okay? The convergence length tells me how long is part of the organ in which I can fit my curvature. So this means that this curvature is supposed to continue up here, but the growth zone is too short to show it, and therefore it doesn't reach the vertical. And the same here. Here the growth zone is much bigger than the length of curvature, right? You see that it's something of this order of magnitude, and therefore you can fit it and you have everything else is straight, okay? So you can understand these dynamics and the final picture just by looking at these different scales. And I think it's very nice that you can relate the two. Yeah?
I'm confused. When all these three have the same length.
Yes, and it's constant.
It's constant. So if I had that guy with a longer length, would it straighten out?
If the growth zone were larger.
No, the low length.
No, if it's just the length, because if it's not in the growth zone, it can't move. So I don't care what happens before. If I have a longer length, okay, this is the current simulation. It's this big. This is my growth zone, okay? So it was like this, and now it can move, and so on. Okay, it's like this. If I add more, but below, where it can't grow, it's like adding like a stick. It can't do anything. So it doesn't change the dynamics that occur up here. Because the curving occurs on everything that grows. If I add a part that can't grow, I'm not adding any curvature. Okay.
Just as a solution to the differential equation that you wrote down,
yeah,
there will be some limits of S that you take, S equal to 0 to S equal to L.
Meaning the base and the apex. Yeah.
Arc length parameter.
Yeah.
If I increase the maximum of the arc length parameter, you're saying that the low B value still stays?
If I increase the growth zone, LGZ, the length that can change, then yes, it will change. But then you're changing.
No, not change the growth. No.
So the only length scale that I'm interested in here is only the area that can change, and that's a growth zone. If I change this so that it will be longer, but that everything grows, then yes, then I will get to the vertical, but that's because I'm changing B, right? Because this will be larger than this. But by adding something which is not in the growth zone, it doesn't change. Apart from maybe the initial angle, depending on how you want to add it, but that's it. Okay, I have 5 minutes, so I'll just quickly say that, okay, that it makes accurate predictions, but maybe this is not so interesting. I want to continue with this because this is, I think, the last slide related to this model. there's one more interesting thing, characterization that we can do to the dynamics. We're talking about overshoots, right? And up until now, we didn't discuss this in this whole model, right? We only discussed the general curving and then the straightening of from the apex downwards, right? But we did see overshoots. So what they did in this work is that they decided to classify different plants by the number of overshoots. So they take a plant, they run an experiment, and throughout the experiment, they count for each snapshot, they look if there are any overshoots, okay? And of course, this changes with time because we know that at the end, everything stabilizes, right, and you're straight. So they're interested in the maximal amount of overshoots. So basically, mode 0 looks like this. So this is like the wheat coloptile, right? It never reaches the vertical anyway. So there are 0 overshoots. Mode 1 is when there is 1 overshoot, as you can see here. This is a snapshot in Arabidopsis. So in all of the movie, this is in this particular example, okay, you have only one time an overshoot. And therefore, this is classified as mode 1. Okay? And in the example of in patients, you can see that in one of the snapshots, you have 2 overshoots. So it has this S shape, and this is mode 2, okay? And mode 3, it would be another overshoot, but they didn't find a plant that has that. They usually stabilize before that, okay? So now they can, for each experiment that they do on whatever plant, they can go and check throughout the video, the time-lapse, which of the snapshots has the most overshoots, and that will determine what is the mode of that specific plant. And what they did is, these are 11 or 12 different plants. You can see that they're extremement varied. You have wheat, bean, sunflower, pea, tomato, chili, raspberry, the impatiens from Pfeffer's experiment, carnation, inflorescence, another mutant, and other data from other papers. So a lot, okay? A lot of very different plants. And each of these, this is the B number that they measured for each single plant. Okay, this is single plant data. I think there are 60 or 70 data points here. And so this is B, and this is the mode, okay? So the green line represents from simulations, when you run simulations for different Bs, what the mode should be. Okay, so it goes, you see that it's a very stepwise function. So below, I think 2.9, you're in a 0 mode, like the wheat. Then you jump at 2.9, you go to mode 1 until 4.1, I think. And then you go up to the second mode, okay? So what you can see is, okay, first of all, there is no plant that displays mode transitions for smaller B than was predicted. Okay, here it's empty. There are no experimental observations here or here, right? What you do see is on the other side, here and here, which is not obvious what's going on here, right? So their explanation for this is probably this has to do with the elongation, which is absolutely not taken into consideration here. And so if there is elongation, it means that you have mistakes in the way that you extract your parameters, the length scales and so on. But there is something to be said that they're able to capture with their model the type of dynamics just from this B function, right? Oh, and we're talking about 11 different species. Okay, so I find this crazy. And it's also quite out of the ordinary in a biological work to look at so many different species and make a comparison. So to finish this up, I just want to do 2 things. Here, I just want to say that if we go back and think of the model requirements that we had, we met them. Okay, so one of them was the simplicity and having a minimal model, right? And this is very minimal. It's super linear. I mean, you see how much stuff we threw away here. And we only have one variable, theta. That's it. And we have only beta and gamma, which are not fitting parameters, right? Because they have a meaning. We know that they're related to characteristic length scales and time scales, which we extract from experiment. And basically you have these 2 terms. We know what the terms are. This is dynamical response. This is the gravity signal, and this is proprioception. Reproduction, yes, we were able to reproduce the dynamics that we mentioned there. And it's also robust and universal in the sense that it's able to describe the dynamics on a large variety of plants, including the poplar tree of Coutin et al., which has a completely different mechanism, right? And I should say that this can also be easily generalized to having multiple stimuli or a stimulus of light, for example, or whatever. And since I'm done, I'll just briefly go through insights. Just look at the titles and then we'll stop. So what we did here is we have one of the main things that we have here is this dimensionless number B, which links the steady state shape with the kinematics. Another interesting thing is that up until this paper, people only used the sign law. So they were only concerned with the fact that you are sensing sensitivity. No one discussed the role of proprioception, okay? Although it was alluded that there is proprioception. And basically what we know is that since we have, for example, with wheat, that it never reaches the vertical, this is something that you can't understand if you don't have proprioception, if you don't use this model. So you cannot predict what will be the final angle, basically, without having proprioception. One very nice result here is that although you have, we said before that small changes of curvature at the base can have big repercussions in the angle at the top, right? And we have all of these overshoots and so on, which are associated with basically what's happening everywhere. But this model tells me that there is no need for some sort of complex regulation where there is a propagating signal throughout the plant. It's enough to just have local sensing and local response. I don't need to know what happens everywhere else. And that's very, very interesting from the point of view of a distributed system in terms of computation. And sorry, the last point. Yes, this worked pretty well while we were neglecting something like, you know, the elephant in the room, although it was raised many times, the role of elongation. We completely disregarded it, okay? And we're saying that it's a driver of movement and so on. And even though we disregard this, we're still able to capture the dynamics with this super simple model, okay? So we will mention next talk, hopefully, why it's okay to do this, in what regimes we can do this, neglect the elongation. But it's, of course, from what we saw with the B model, right, with the modes, it's obvious that that's the next step, okay? And this, of course, is what we'll do next talk tomorrow. That's it. Thanks. Any questions?
So if we were to write an analogous mechanical model rather than a geometrical one, this is a very geometrical model. So what kind of meaning would you ascribe to beta and gamma in terms of some mechanical properties of trees?
So I wouldn't. There are separate things that you can add on top of the other, but you can't disregard them. Actually, a very nice way of putting it is this. When I was talking about the response to gravity, I think I mentioned somewhere in one of the titles as opposed to the passive response to gravity. Mechanics tell me that if I put something like this, it droops down, right? This is the mechanics of gravity. But what I see is going up. This is the active response to gravity. And it's two separate things. One of the interesting things to do is, of course, to add mechanics, because you see, if you look at plants, you see there is a variety of plants, right? You have these drooping plants where they fall down. This is due to mechanics. But then at the tip, where the remaining material is not so heavy, they go up again. Okay, so there's an interplay between the active response to gravity and the mechanical response to gravity, but you can't replace one with the other. It's one on top of the other. Yeah.
It's related to something like the response time of these plants, in the sense they're searching for something, and somebody moves. It's like one plant has like a long strides, so it takes steps and misses, then it actually has to come back again. On the other end, there are plants that respond like, slowly, so they can actually, this is pretty much happens when you're searching for a parameter, and it depends on how finely you tune.
Well, you can, to some extent, say maybe...
So the beta, is it sort of related to like, in the sense what I mean is like, if you do an experiment, let's say, where you keep like, changing the orientation regularly. So after, there might be duration where if you do it very quickly, they will stop ignoring. I mean, like it's not enough time for them to respond to the switching.
Yes.
And there's a time when they respond. And there is a sweet spot where it's actually in the same scale as the response time of the plant. So could that be related to what causes these modes in some sense?
You're saying whether these oscillations or lack thereof are due to somehow the response time, the internal... So let's think out loud. The response time of the sedimentation, so this is at the cellular level, the lowest time scale, it's about 3 minutes. So if you do anything, if you make changes under 3 minutes, it shouldn't respond to that. But the growth is, no, the rate of growth is also different between plants. I would say that it's no. Beta and gamma tell you how sensitive you are to something, okay? Such as, let's leave proprioception and look just at the model with gravity, okay? So you'll see plants that respond, or light, there is more data to that. You see some plants that will respond very quickly to gravity, and other ones will respond less. Not only quickly, less. The maximal angle will be less. You know what, that's a good point. Now that I think of it, maybe it is related to elongation rate, because what allows the bending is driven by the difference in growth rates. So if in one type of plant, the difference between elongation rates can be physiologically, it's so that it can be larger, it means that it allows you to be more sensitive to a stimulus. You can go to a greater curvature. And in fact, you can also define from these numbers what is the maximal curvature that the plant can attain in the first place. So in that sense, maybe yes. So it's a yes or no answer, not helping a lot.
Sorry, I have a hard time about the final plot when you have 3 modes.
Yeah.
Because the 3 modes are 3 categories, like discrete categories that were defined, but like how did they fit the real data into 3 categories? Because I'm assuming there was certain variation in real data. It was not absolutely...
No, each dot there is one plant. So you'll see that you have a few of wheat, for example, and there you see that there is actually... wait, what did I do? Sorry about that. Okay, for example, okay, the red dot, that's wheat. You have a lot down here, but you have also some up here. Okay, so there are some, although it's exactly the same type of plant, most of them will be on the 0, okay? They have no overshoot, they're this type, but a few of them do overshoot. So, but it is absolute for each, because for each experiment, I look at the time series, and I just look where do I have, if any, most overshoots, and that's it. So it's a binary or trinary. No, there can't be. It either overshoots or it doesn't. What's a half overshoot? There is no such thing.
Okay, fine. So if not, let's thank Yasmine for a good talk. We take a coffee break and come back at 4.